Neurological Disorders, Lesson 5.1, What Circuit do Drugs Affect in Our Brains? The goals of this lesson are to introduce students to the concept of addiction and to introduce the neural reward pathway. At the end of this lesson, students should be able to compare and contrast human and rat brains, describe how scientists study addiction, and name the parts of the brain involved in addictive behaviors. We'll get to these goals with a Socratic discussion and graphing and analyzing data. To prepare for this lesson, you'll need to review the key scientific concepts presented in it. They include addiction, which we'll define as the continued compulsive use of drugs despite adverse health and social consequences. The methods of studying addiction using animals, including intracranial self-stimulation and self-administration. And finally, that drugs hijack the brain's reward pathway, which includes the ventral tegmental area, or VTA, the nucleus accumbens, or NAC, and the prefrontal cortex. You can review the scientific content in the background reading provided to you in the teacher primer, the teacher manual, and the student workbook. The teacher primer provides in-depth knowledge about the scientific content presented in the lesson. The teacher manual or lesson plan provides a minute-by-minute explanation of lesson structure, including instructions on how to manage the discussion and activity, and the student workbook provides additional explanations for your students. You'll need to be sure to print the activity background worksheet, the activity worksheet, and the homework worksheet for your students. The key points of lesson Do Now is to engage students with the concept of addiction, as it is the example we'll use throughout Unit 5 to discuss how our choices change our brains, and to pull together all of the previous units about the brain, our neurons, our synapses, and our circuits. Additionally, the Do Now serves as a formative assessment of your students' understanding of the ways to study addiction. We'll get to these key points with a peer-to-peer discussion. In the Do Now, students work with a partner to define addiction and brainstorm ways in which scientists could figure out what parts of the brain are involved in addiction. We'll discuss the Do Now within the lesson's discussion. The key points of the discussion are to first introduce the use of animals in scientific research and to explore the similarities and differences between the human and rat brain, specifically that the human brain has a more developed cerebral cortex and that lower brain structures are almost identical between the human and rat brain. We'll get to these key points with a Socratic discussion. The discussion starts by reviewing the Do Now with students, and if students haven't already gotten there, pointing to the idea of using animals as models of human behavior. Since animal research is a very important component to all medical sciences and one that does raise some ethical concerns, it is an issue we'll revisit in Lesson 5.3. But for now, the focus is that animals serve as an important tool for scientific research. Slide 4 allows the class to compare the human and rat brain. By comparing the brains, students should notice that the human brain has more developed cerebral cortex, but that both brains' lower structures are very similar. These lower structures are the ones that govern addiction, which is why animals can be a useful model to study the very complicated human disease of addiction. The key point of the lesson activity is that drugs hijack the brain's reward pathway, which includes the ventral tegmental area, the nucleus accumbens, and the prefrontal cortex. We'll get to these key points by graphing and analyzing data. In the activity, students will complete a lab on paper to answer the question, how do scientists figure out what parts of the brain are involved in addiction? You'll give each student a copy of the activity worksheet, which instructs them to graph and analyze data from experiments with rats. For these experiments, rats were placed into individual cages with 2 levers that the rats could press. If the rat pressed one lever, it received food. If a rat pressed a second lever, it received either an injection or an electrical stimulus applied directly to its brain. Working in groups, students will graph the data for 4 different rats, A, B, C, and D, and after graphing their data, will use their worksheets to analyze and interpret this data. The key point of the second discussion is again that drugs hijack the brain's reward pathway, which includes the ventral tegmental area, the nucleus accumbens, and the prefrontal cortex. And we'll get to these key points with a Socratic discussion. After the students have finished the activity, the next set of PowerPoint slides allow you to facilitate a discussion about the experiment. This slide allows you to present the data students should have graphed during the activity. You could also have students draw this data on the board. Either way, you'll want to have a presentation of the data readily available for the discussion to be able to reference it and guide students to appropriate conclusions when evaluating it. Key questions that you'll ask the students to help them analyze the data include: which rats press the stimulus lever the most? which rats pressed the food lever the most, and then which rats displayed addictive behaviors and why or how do you know that? After comparing the behavior of the 4 rats, you'll reveal to the students what was happening within the rats' brains after they pressed the stimulus lever. Rats A and B were receiving an injection, whereas rats C and D were receiving electrical stimulation. You'll then ask the students which rats displayed addictive behaviors. Both rats A and C did, whereas rats B and D did not. From there, you'll compare what's similar about rats A and C and different from B and D. Rats A and C both have activation of a specific pathway. This then allows you to introduce that pathway to your students. This pathway is the brain's reward circuit and is composed of the ventral tegmental area, the nucleus accumbens, and prefrontal cortex. This pathway is conserved between humans and rodents, and scientists have learned that all drugs of abuse, and in fact all pleasurable behaviors, activate this pathway, which is why it's called the reward pathway. The key point of the wrap-up is that humans abuse drugs because they, the drugs, activate the reward pathway and cause feelings of pleasure. We'll get to this key point with a reflection and discussion. In the wrap-up, we'll address the question of why do humans abuse drugs? Yes, this is a loaded question, and one that we'll continue to build on as the unit progresses. But the goal for today's lesson is to acknowledge that humans abuse drugs because they, the drugs, activate the reward pathway and cause feelings of pleasure. Feeling pleasure feels good, so we repeat whatever it was that made us feel good, which here is using and abusing drugs. The key point of the homework is that addictive behaviors activate the reward pathway. We'll get to this key point with a homework worksheet. The worksheet includes a reading and questions about addictive behaviors, which in the study highlighted includes drinking chocolate milkshakes and how they activate the reward pathway. Students often ask for more details about addiction, which is great because this entire unit is about addiction. So you can find more in the teacher primer, the teacher manual, and the student workbook. One of the most common questions teachers get is, if all things that make us feel good activate the reward pathway, why don't we get addicted to everything? The answer to this question rests on the idea that the level of activation of the reward pathway is critical in determining addiction potential. Drugs of abuse will activate this pathway 10 times or more than other pleasurable things, like chocolate milkshakes, spending time with a friend, or winning a game. At the end of this lesson, students should appreciate that our reward pathway is activated by all drugs of abuse, and that this pathway is well conserved between humans and rodents, which makes rodents a good model to study the science of human addiction. This lesson builds on students' understanding of neural circuits by giving them another example of a circuit, also known as a pathway, involved in controlling behavior. This lesson also lays the foundation for remaining lessons in the unit that use addiction to pull together the entire module and demonstrate how our choices change our neurons, our synapses, our circuits, and in the end, our brains. Don't forget, if you have any questions, concerns, or feedback, to let us know. You can contact any of the CTSE team members, and we'd be more than happy to help you.